Hello everyone and welcome to the HPTLC Online User Meeting. My name is Maricel Marin-Kuan and I am Senior Scientist at Nestlé Research in Lausanne, Switzerland. I will address today how to assess the safety of food products in absence of toxicological data. During the presentation, we will address the following points: the safety assessment in the food context, the hazard identification in absence of toxicological data, the contribution of bioassays and alternative methods, such as HPTLC, and some conclusions and take-home messages. The context of this seminar is the chemical food safety. In order to assess the safety of chemicals present in food, we need to consider the following parameters: the toxicology, which refers to the hazard identification using chemical analysis, and the hazard characterization using published data; the exposure, which refers to the food intake and the occurrence of chemicals in the population. Together with the toxicology data and the exposure data, we can perform the risk characterization in order to provide a recommendation for decision-making and prioritization. However, in absence of this toxicological data, we cannot address the safety of chemicals. This is particularly relevant in the food context, as food and food-related samples are actually a mixture of chemicals. Let's consider one example with the packaging material. Many chemicals are implicated in many packagings, potentially migrated into food. And the question is how we can address the safety. If we perform a migration experiment with packaging material, we will observe with the GC-MS analysis that the migrate contains known-uncharacterized substances, but also several unknown and unexpected ones. From the toxicological perspective, we will expect to fully identify and characterize these substances, but of course, this is unrealistic. And we strongly believe that the bioassays can play an important role to exclude endpoint of toxicological concern to allow the assessment of the safety of complex mixtures. For that, we have developed a framework to bring breakthrough solutions to the safety of mixtures using bioassays approaches. By selecting toxicologically relevant targets, implementing and applying state-of-the-art screening tools, building know-how competences with external research collaboration, and assessing efficiently for prioritization and decision-making. We have identified two main toxicological endpoints: the genetic damage and the endocrine active substances. The genetic damage, for obvious reasons, as stated also by the European Food Safety Agency, due to the adverse consequences of genetic damage to human health, the assessment of mutagenic potential is a basic component of chemical risk assessment. And endocrine substances, because these substances can interact or interfere with normal hormonal action, but may or may not cause harm. This topic is actually of increasing concern in regulation and is of poor public perception. Therefore, we consider that the bioassays to assess the effect on DNA damage and on the endocrine system are relevant and need to be taken into consideration. Therefore, we decided to set up a battery of tests to assess those toxicological endpoints. For the genetic damage, we set up a bacterial test, which is called the AIMS-MPF assay, which is recommended to assess the mutagenicity. For the endocrine activity, we set up a human recombinant cell lines system, which is called the CALUX assay. Here, we can observe an example for the estrogen receptor. We can assess the activation or the inhibition for the different hormone receptors. Despite the performance of these methods, we observed some limitations according to the application field we needed. We observed that the limit of detection is not suitable to exclude mutagenicity, that the cells are very sensitive to solvents, so that limit the exposure concentration. The wall extract exposure can be a problem. We can observe matrix effect we have no clue about the molecule responsible of the biological activity and is both time and material consuming. Therefore, we decided to implement a new in vitro competence to analyze complex mixtures in our laboratory. So we implemented the high-performance thin-layer chromatography with the following stages. The stationary phase, we are using the normal phase silica 60 plates because we have found much better sensitivity using these plates as compared to the reverse phase. For the samples, we are using extracts or compounds and always the reference compounds needed for each of the bioassays that I will show you afterwards. Usually our samples are dissolved in ethanol. For the mobile phase, we were using mobile phase adapted for each sample under analysis, but now we are using a 3-mobile phase concept that was developed by CAMAC, which is much better because we are really assessing all the different selectivity according to the polarity of the samples. For the bioassays, we have coupled 2 bioassays, one with a bacterial system and one with a yeast system. For the bacterial system is the SOS Humoxy assay, which is a test that is commercially available and is used by for environmental sample analysis usually. This test is based on the Humoxy gene in bacteria that is only induced when there is genotoxicity damage. It's very important to assess this test in presence and absence of metabolic activation because we need to assess not only the parent compounds responsible of the genotoxic activity, but also the metabolites. And for the yeast estrogen activity screening test, the YES test is also a commercially available test that is also used for environmental samples for estrogenic activity assessment or other samples. So these two tests were implemented for the assessment of the genotoxicity or the estrogenic activity. Here we see an example of a dose response test for the Humoxy test or the YES test with a dose response effect. Very nicely we see the signals with the different fluorophores that need to be used with each of the bioassays. FDG with the Humoxy to avoid interference with autofluorescence, and the MUCT fluorophore for the YES test. We can quantify it and observe the dose response effect as a standard test. And very importantly, our next step is to confirm the chemical identification of the molecule responsible of this activity for the genotoxicity or for the estrogenic activity. For that, we recover the bioactive bands by applying 15 times in the silica plates. We recover manually or with the interface device, and we do a short extraction to eliminate the silica, and we analyze the extracted samples for the chemical analysis to try to identify the responsible of the activity. And we can use also this extract for the bioassays that I showed you before, for the EMS-MPF test, for the Calux test, or to test it back with the HPTLC test. This is a very brief explanation of the protocols, but you can find the protocols for the SOS Humoxy test in the manuscript that we have published in collaboration with Professor Morlock, that we were able to demonstrate with a proof of concept that the method is working for the assessment of the genotoxicity. We are submitting another paper where we are showing the introduction of the metabolic activation in the test, as I explained you before. So we have the proof of concept and the demonstration of the reliability of the test with a set of genotoxic compounds. So now this sensitive planar SOS Humoxy assay it's available to assess the genotoxicity in the laboratory. But I need to clarify it in one concept, because as you saw before, we are talking about genotoxicity and mutagenicity. Just for clarification, let's discuss about what is the difference. When we are talking about DNA damage, we can have direct damage or indirect damage. What does it mean? If we have a direct damage, that means that a molecule can damage directly the DNA molecule, and we are talking about a mutagenic effect. And for this type of damage, we have no threshold mode of action, so it's more severe. And the way to address the assessment of the mutagenicity is with the Ames bacterial test, which is a standard test. By the contrary, when we have an indirect effect, that means that there is a reaction that happens before there is a DNA damage, and it's this indirect damage to the DNA molecule. This is called genotoxic, and for this we can talk about a threshold mode of action. So we can have a repairing eventually. The SOS-UMUXI test can detect both mutagenic damage and genotoxic damage. So we don't have this specificity, but we know that we can address both. So I will show you now some food case studies applications of this HPTLC method that we set up in our laboratory. One using a packaging material, a paper material, and one using a food ingredient, the soy isolates. So we use the paper-based material to evaluate the feasibility of the approach that we developed to go from the extract or migrate of packaging to try to identify the molecule responsible of genotoxic or mutagenic activity using the paper as case study. So here we observed the paper material that we used, and we did a hexane acetone extraction from a commercially available paper material, and we did also the extraction using a spiking approach. This spiking approach was done in order to assess the feasibility to detect a genotoxic compound. We use the 4-nitroquinoline 1-oxide, the 4-NQO, which is a reference genotoxic compound known to induce a mutagenic effect. So we did this, prepared these two samples, and as I mentioned before, we applied in normal silica plates, and we applied the 3-mobile phase concept with a low, middle, and high polarity in order to broaden the selectivity of the samples. We applied the Salmonella SOS-UMUXI assay in presence and absence of metabolic activation, and we detect the molecules, if there were molecules inducing the SOS-UMUXI gene. Then, when we observe the profiling of the molecules present without metabolic activation or with metabolic activation, we recover those samples, as I described before, and we extracted the bands to test with the EMS MPF test to evaluate the genotoxicity, to confirm that we are able to recover the spike molecule, and as well the chemical analysis using LC-HRMS to try to identify the molecules responsible of this activity. So for the proof of feasibility of the HPTLC using the spiked sample, we were able to detect the mutagenic activity of the 4-NQO with the HPTLC SOS-UMUXI test, as we see here the signal with the fluorophore FDG. We recovered this band, and we performed mutagenicity and chemical identification evaluation. For the mutagenicity with the EMS-MPF, we observed that we were able to recover the mutagenic effect of the molecule 4-NQO, as we see here an induction of the revertants present in the EMS-MPF test. And with the chemical analysis, we were able to observe the peak of the 4-NQO with LC-HRMS analysis. So we were able to demonstrate the proof of feasibility of the HPTLC UMUXI method with this spiking experiment. And for the paper sample analysis, I will show you one example with the mobile phase 2, with which we observed two bioactive bands at RF045 and RF015. We did the chemical analysis and we were able to identify two compounds, the abietic acid and the dihydroabietic acid. We learned from the literature that the abietic acid is reported as a genotoxic and direct mechanism compound. So we did a confirmation of these compounds with the HPTLC. And we observed that if we see here the paper-based extract from the beginning and the two compounds, the RF was in alignment with what we observed with the bioactive bands. So we were able to confirm with high confidence that the abietic and the dihydroabietic acids are present in the paper extracts. We did also the AIMS-MPF test and we observed that there is no mutagenic effect with these two compounds, confirming what was described in the literature as genotoxic but not mutagenic compound. And therefore, we can conclude that this approach, with the proof of concept with the 4-NQO and with the extract analysis, that this approach contributes for prioritization of new packaging material. And now we can go through the endocrine activity case study. As mentioned before, the endocrine activity substances can modify the hormone system function, but may or may not cause harm. These substances can be extrinsically or naturally present in food, like the case of the isoflavones. There are several chemical analysis and in vitro assays that have been developed to identify the endocrine activity in food and plant-based foods. However, the consistency between the chemical analysis and the endocrine activity need to be addressed in order to properly address the safety. Here is an example of the ligand test that I show you with the in vitro test. We can have the activity of the estrogen receptor, androgen receptor, and we can have a total activity of the sample that we are analyzing, but it would be very important to identify the compound responsible of this activity in order to address the safety. And we applied the HPTLC effect direct analysis, as we described before, with the chemical analysis to facilitate the characterization of estrogen activity present in food. The soy isolates samples were analyzed to assess the estrogenic activity, as I described before. And here we can see the results. So we did the extraction of soy isolates using a Quetscher standard method. And here we observed the HPTLC profiling of the soy extracts. So we did two different extracts of the soy isolates, and we did a revelation with MP-PEG to observe the flavonoids. So we nicely observed the different flavonoids present in the soy extract. And we did a revelation at 254, which is recommended to also identify isoflavones that we knew are present in the soy extracts. So for the estrogenic activity with the PES test, the two extracts show a very nice dose-response effect with the induction of the MUC fluorophore, and the quantification of the activity was done in comparison to the estradiol reference compound that we also do, of course, with the HPTLC. And here we observed that there was a very nice sigmoid curve for the induction of the estrogen activity present in soy isolates. So we were able to really confirm with the HPTLC yes test that the estrogenic activity that is expected in the soy isolates was well detected. The next step was the identification of the compounds responsible of the estrogenic activity. For that, we recovered the bioactive bands of the estrogenic activity, which were corresponding to two bioactive bands at the RF of 0.6 and RF of 0.4. So we recover these bioactive bands for chemical identification using LC-HRMS. And we observed several candidates, and here I'm showing you the 5 main candidates that we observed: the genistein, the dietzein, the glycitein, the naringerin, and finally the formononetine. These compounds are described to be present in the soy and are responsible of the estrogenic activity, and we test each of the individual compounds with the HPTLC PES test, and we were able to confirm the estrogenic activity present in these compounds. Then we did a concordance analysis with an interpolation according to the amount of estrogenic activity present in the compounds and the amount of this compound present with the LC-HRMS, and we were able to conclude that the genistein and the dietzein are the main responsible of the estrogenic activity. We are publishing these results and we expect to submit this paper the next weeks. With these two case studies that I presented, I hope it was clear for you that we have successfully demonstrated the potential of HPTLC to the contribution of the biodetection as an approach to characterize complex mixture. Actually, we successfully integrate a highly sensitive bioassays to our existing battery of assays. We have integrated the chemical analysis, which is a very critical step for the safety assessment of chemical compounds present in food. And we can facilitate the risk assessment as a way forward for the safety assessment of complex mixture. Just as an information, we have also an in silico technology included in the battery, because this is really key for the prediction of potential toxicological concern. So finally, we arrived to the conclusion and the take-home messages of our seminar. We conclude that we did a proof of concept of an approach bridging effect direct bioassays with chemical analytics to qualitatively and quantitatively characterize genotoxic and estrogenic compounds present in food, like I show you with the packaging material and the food ingredients. That, very importantly, HPTLC can overcome some limitations of the multi-well plates with good detection limit, reliability, versatility, and efficiently. And we can have another advantage that we didn't mention, but is the semi-automation using the HPTLC approach that we have now in-house, and it's very performant for the bioassays as well. We have an important contribution for the characterization of food and food-related samples, which is definitely facilitate the safety assessment. And this is really, for us, the main advantage of the HPTLC. Of course, we would like to expand the HPTLC application with the implementation of new biological and chemical derivatization methods that are already ongoing in the laboratory or in collaboration with other stakeholders. If we address the question if high-performance thin-layer chromatography is a suitable approach to evaluate the safety of complex mixtures, the answer is definitely yes, because we observed several advantages: the versatility with a stationary phase, the mobile phase selectivity with the different polarities, the derivatization protocols that are already available. The sample preparation is facilitated. There is less or no solvent limitations. We observed that the limit of detection can be improved as compared to standard methods, as we demonstrate with the HPTLC UMOXY test to exclude mutagenicity and to be in alignment with regulatory requirements. The fractionation, not only to diminish the matrix effect, but to target to the bioactive band detection versus whole sample signal evaluation. And this facilitates the identification of the molecule responsible of the biological activity. And of course, this feature is the powerful edge of HPTLC because this facilitates the assessment of the safety. And last but not least, with the semi-automation possibility with the HPTLC Pro, we can program the HPTLC bioassays the day before, and in the morning we can see that the samples are applied, they have the chromatography, and we can apply the bioassays the next day. And we can perform much more samples as compared to the manual test. And we, of course, we have an advantage of the time and the consumables with the HPTLC. But we have also some disadvantages. We have only few HPTLC coupled bioassays available, so we need to work on it. Mammalian cells with which we have a lot of possibilities of methods. We cannot test with the HPTLC because of survival of the cells problems. We need to improve the cytotoxicity assessment with the HPTLC in the same plate than the bioassays we are performing. This is critical for the performance of the test. The qualification and harmonization with the HPTLC in order to allow the validation and recognition by regulatory bodies. And with this, we arrived to the end of this seminar. And of course, this great adventure to implement the HPTLC technology at Nestlé wouldn't have been possible without a great team. And I want to thank the biodetection group at Nestlé, with whom we are working hand by hand every day to implement and apply this technology. To Benoit Schilter, with whom we started this idea to implement HPTLC at Nestlé. The packaging food safety group, with whom we are applying for the packaging material, the untargeted screening group, with whom we are applying for the plant-based samples. But all that wouldn't have been possible without the great support, training, coaching, and patience that the experts in this field have provided to us, particularly Professor Morlock and Pierre Savary, with whom we started this adventure, and the precious support from CAMAC that is really ensuring that we succeed with this initiative, and to many other stakeholders. So with that, I want to thank you a lot for your attention. I provide a lot of information, so please don't hesitate to contact me to my email address if you have particular questions or doubts, and I can try to answer to you. And now we open the discussion for the questions you may have to finalize our seminar today. Thanks a lot.